Hi guys, and welcome to our entry-level fluorescent microscope. So it's this microscope is in the second floor, room 255, together with a confocal microscope and a turf microscope. So as always, when you start out, you need to sign in the logbook, just for the next user to see if you had any problems. And the startup procedure is written on the wall, and this is what I will show you in this short video. And also you see the shutdown procedure down here. Moving down, you see the spectre of the light source and the filter sets that we have in the system. So the light source has a UV peak where we detect the Höchst stain. Then it has the green diode where we detect the FIT-C. And then the red and the far red you see out here. So it's designed for the Sci 3 and the Sci 5. This is controlled in the software already. So I'll just start out showing you how to start it up, and that's quite simple. In this case, we just turn on the power. So turning on all the components. And then this guy is a touchpad controller controlling the microscope. So that's a basic way of starting it up. You can use it this way and simply analyze your samples, but mostly you would like to start out imaging right away, so you also turn on the computer. And I can introduce you to the components. So we have in this microscope four lenses right now, so it's 10, 20, 40 and 60x objectives, and actually also 100x objectives, so we can cover the whole range. And the dyes we can analyze is Höchst, FIT-C, Sci 3 and Sci 5. And you can see here the stage where you insert your samples. So right now I just put in a sample, slide it in, and with my right thumb putting it all the way in. And you can fit one more if you need to. Then you can move the microscope to wherever manually, and you can operate it by the touchpad. You can use full operation mode if you're not using the computer. Here you can go up and down with the set, the focus, and you can move the stage in this part, and you can also control everything here. Here you see the upright microscope, and you have transmitted light from below, and you have fluorescent light from above. You can see the white light source over there. Moving down here, you see a control of the stage, so a mechanical joystick over here, and you can control it either in coarse or fine mode. The same goes for the set drive. You can have fine movements of the set and coarse movement of the set here. Also the fluorescent shutter and functions that you also have in the touchpad control you can see here. When you operate the microscope manually, you will use the splitter up here fully in, so then meaning you have all the light to the oculars. When you take it all the way out, then you see everything with the microscope camera. And this is a sensitive camera, black and white camera with a quantum efficiency of approximately 60%. And you want to use all the light, so you want to take the splitter all the way out. Okay, now I will control the microscope from the touchpad control. And first you see the different objectives here. So by touching those, it of course changes objective. And now you can see over here the set. It's going to the focus position, and you can control it here. So you can see the pointer going up and down again. And when you close your specimen, you're getting close, and it will lock, so you're not going to damage your objective. But when you are at this point, you can find focus here or on the focus wheel on the other side of the microscope. You can control the stage by moving over here with the coarse movements, or touch this one and move with the fine movements. So that was the first tab here with the XY stage. Then we can go to Epi, which is Epifluorescence. So we are in the tab called Fluorescence anyway, which is a system is made for. And you can here select the different dyes. So this is the filter cube for Höcht, and you have also EGFP, M-Cherry and FIT-C. So you can also go to Brightfield observation if you want to see the tissue in Brightfield, and you can then control the transmitted light. So this LED you can control in here, and you can also control the iris, so the aperture stop and the field stop to make better contrast. So I will find my sample. I have now found focus, and I will move around using the XY stage. I can either do that manually, or I can do it on the touchpad here, or on the manual switches on the side of the microscope. And once I found myself, I can press set. Then maybe I want to go to oil objective. So I'm now at 10x magnification, and I want to go to 60x using oil. So I have my area of interest in focus right now, and then I press set on the touchpad control. Then I can manually take out the stage, and I can add a drop of oil. And then I press go, and it will find the same position again. Then I change to 60x oil lens, and I'm in the position I chose before. So if you want to control the microscope manually, then you need also manually to control the light source. That is, first of all, turning it on. When it's turned on, then you need to select wavelength. So this is the UV diode, and it has UV, and it has a blue color and a green-red color. So the UV is for Höcht, and the blue is for FIT-C, and the green-red is for Psi 3 and Psi 5. And you select simply the wavelength you want and the intensity you need. And in most cases you don't need very high intensities. If you use the software, then it's controlled everything here in the software. And now I will turn on the computer to be able to control everything from the software. So the only thing you need is these three icons. So the cell sense dimension is the software we control the microscope, and the other ones are just for mapping our drives. This is the start page, and you start out in the camera control, where you can see live and snapshot. In the top part you can see the different objectives and the shutters, so the transmitted and the reflected shutter, and also the different dyes, GFP, Höcht, M-Cherry, Psi 5. And we are in the top panel. Acquisition is the main part on this video, and then you can see processing and count and measure is something you do after acquiring, and then it has a reporting function, and to the very right upper part you can go in and see the layout functions, simply a way of displaying the software. Now I shut down some important features, and when I go to layout and reset current setup, I see all the functions again. Here you see the microscope control, you can go to the right corner and control the XY movements. And you can also control the Z, the focus movements, and all the filter cubes, the LED, so the transmitted light percentage, and the condensers, and all what the components of the microscope. Down here you see 3 diodes of the fluorescent lamp, so UV, the blue, and the red. You can see when I change to the red, it uses this green and red light diode. Here it's showing Höcht, so it's using the UV light. And I see low magnification, 4x of my cells. And when I click to the right side of the software again, I see my histogram of, so I can adjust the display. And using autocorrection, it clips 0.1% of the darkest and the brightest areas, so I see a good signal. To the left, you can control the stage. You can go to Stage Navigator, and when you hit the first button, you're defining the overview, and you are asked to go from the upper left corner to the lower right corner. Now it's defined. And the next button to press is Acquire Overview Function, and you see the software acquires all the different components. So this was both slides in the low magnification, and one of the slides is showing a very bright intensity in the lower right corner. But the purpose of this is to have an overview to be able to go through very fast through the sample and find some interesting parts. Now I go to 40x, so I go from 4 to 40x, and you see the square, the yellow square is much reduced. When I go to 40x, the focus needs to be readjusted, and you see a lot of expression because you are getting too much intensity with the same exposure, so I take down the exposure time. And I change the display. You see the blue is showing darkness, and red was showing overexposure. And by pressing Control H or the saturation indicator up here, you activate the pseudo color, the blue and the red colors. Next function is autofocus. You can autofocus on the whole region or on a part of the region. So this is the whole region, and it's doing autofocus in the lower magnification. Now I move to another position and go to 20x. And I press Control and the mouse wheel to focus. It's often faster than doing autofocus. Going to 60x oil, we need to add oil, of course, to the sample. And then we get a very good signal and high intensity. Now I'm adding positions, so I click this Add Position in State Navigator. you saw just here it's adding positions. So I'm adding a rectangle, so I'm moving from top left to lower right. So I think this is interesting, so I go to the top left, lower right, and I press finish, and it will acquire all these points within this region of interest. And that is 12 images stitched together. I go to position list, and then I see my position 1 is this tile of 12 images. Then we want to update the focus, so I go to a position and readjust the focus and press update C. Then I can go to another position. Yeah, I go somewhere else. And you can add a circle. Or simply a position like I did here. Now I'm messing things a bit up. Adding positions on top of each other. This was a circle. And going to the position list, I can just delete the ones I was putting on top of each other. I press go to position in the center of this circle of the many positions, and I'm updating the C, the focus again. Then I'm going to Process Manager, adding multi-positions. Once I add this multi-position and press acquire, or it will go through my 2 positions. And it will take some seconds on this, or some minutes, depending on the areas, and the small position here and the big position with the numerous images. Next function is a line. You can add a line profile to the image. This one is important, the acquisition setting. So you can go to the document name, name the snapshots. That is images taken in the camera icon in the top. And give it some meaningful name here. And here you can see it's counting. So it'll start from 1 to 3 and so on. The next one is process. And I'll show you that in saving also. So the first is snapshot. You save snapshots separately, and you save then the process or experiments as other types of file. So found your local drive, or you can add it directly on the network. I just put it into my local drive here. And after acquisition, I move everything to the network drive. This is done for snapshot, and then this is process. So that's meaning multicolor, C-stacks, whatever you do that is more advanced than just simple images. And you can give it names, like you have probably used to from other software, add it or remove it. And you can make your own, modify it, and give it some special name if it's not in the list to the left. Now I activate also multicolor and C-stacks, so you can see the activated icons are blue. Then I have my Höcht and my Sci5 channels. So double click, you go to the other channel. So now I double click on the Sci5 and see some structural proteins. You see here the saturation indicator. It's not saturated, so now it's saturated. You want to avoid that, and you see it also in the top right where you see the histogram. So back to Process Manager. Once you have found your exposure time and your settings, you go to Read Settings, so the Process Manager knows the exposure times, etc. I just make a clean appearance here, so closing all the files. I press Live again, and I want to acquire 2 channels with numerous set planes. So going to the Set tab, set the top and the bottom positions, and press Apply. And then we press Start, and it will go through the 2 channels and a number of C-stacks. And you can see it's going through it here. And this is important to click C before channel. If you don't click activate that, it will go from blue to red and then go 1 stack down and switch filter cube every time, and that will increase the time a lot. So now it's done. You can see the display here going through the different C planes up in the top. Now we have only C to change. If you had time, you could also go from to show different parameters. Here we go, automatic, like making a video through the C, and show it different ways. So you can also show it in this ortho view. You can go through the XY and see the depth in the right and the lower part. And you can make a 3D, which is quite thin with this sample. You can also add positions, so if you want to make multicolor C-stacks at the different positions which we went through before, then we can add several positions. And here we add 12 positions, or 12 images in tiled together in one position. And of course it will take some time to be done with that. You can see some of the first images here. In the lower part you can see the estimated time of 2 minutes something. So these are 2 first images. And now it's done. Here we see a tile image in 3 dimensions. And this was our previous image. And you can see it's tiling quite well. You don't see the borders. You can also add a time lapse. It may not be very much used on this microscope because it doesn't have incubation, but you may have reasons to make a cycle of it and go through the positions or whatever kind of protocol you want to make. So this is exactly the same image acquired the time-lapse loop. You can see here T is representing the time, so we have different channels into. Now we go to the other image we acquired just before. Again, let's activate C-stacks. Finish the time, so you can define the time, or you can make it as fast as possible. So now we see the different set layers and we see the time. Of course the image looks the same. Let's move to our second slide. You can see in the stage navigator to the left, the square was moving from the top to the bottom of the position. So this is a different slide showing some more colors. So it has M-cherry. And activating M-cherry, you see now I set the exposure time. And you see the display in the histogram. It's a quite old sample, so it's not the highest intensity. I'll set my top and my bottom again, so press set and then apply. And then we are acquiring 3 colors instead of 2, what we did just before. And this is just a test. The signal in the M-cherry and Sci5 is pretty much the same. I'll just show you this so you are able to do 3-color imaging. So now we are done imaging, and we want to leave the system clean for the next user. So first of all, you go to escape position, and we need to take out the oil. So you can press this button to go all the way up, so you can have your working space cleaning the lens. Take out the sample, and with some ethanol here, you first clean the sample. Circular movements, of course, take care that you're not damaging your sample. In this case it's sealed, so it's not important for me. And you take a new piece of paper, and then you wipe the lens with circular movements, something like this. And then wipe it with a gentle hand with a piece of the dry part. So now it's clean, and we're ready to shut down the software. You can just as well shut down the computer at the same time. Then you see that still the touchpad is active, and we can shut it down manually on the back side right here. And the last thing you need to do is to power off the whole system and cover it.